My name is Zach Chandler, and I'm going to be covering the inheritance patterns of molecular biology diseases. An overview of what's going to be covered is going to focus on autosomal dominant and two recessive genetic disorders and their pattern of inheritance to show how this comes about. It's best to start by focusing first on DNA structure to provide an overview of the double helix, which is going to be the smallest unit where the DNA code is written, and we will expand it all the way up to a karyotype that contains all the chromosomes that contain all this DNA information. Next, I'll focus on the patterns of inheritance and the pedigrees, which are ways to track genetic diseases through families. And the examples I will cover will be genetic diseases that will be dominant for the Huntington's disease, and I'll focus more on the recessive diseases, cystic fibrosis, and sickle cell disease, and I'm also going to cover their location inside the genome. This slide provides a good overview of the DNA and its structures. Starting at the smallest gene sequence, we see that we have these bases here, which we'll look into more. And at the smallest level, the DNA is unwound and it can be translated or transcribed into proteins. And at a higher level, these double helixes become more wound tightly, and this is thanks to proteins such as these histones that keep the DNA wound very, very tightly, and it continues on and on until you get to the size of a full chromosome. So you can expect only particular regions that are being expressed often will be in this open format, whereas when they're not being expressed, they'll be more condensed and closed at the larger chromosome level. So it's important to know the central dogma of biology. This is not 100% true, but it's generally how most things operate. So DNA is contained in many living organisms. It will be able to transcribe RNA that will have a single strand and will have a genetic code on it. And this can be translated in groups of threes, which matters from the 5 to the 3' direction. And the way those are read in groups of threes, known as codons, will determine the amino acid that is created. And multiple amino acids will build up to make proteins. So the mRNA's codon, you can see is down here, the UAC, which mimics the strand that we see here in DNA, but instead of having a thymine, RNA has a uracil. So like I said, the direction matters from 5 to 3', so we'll look at UAC. So come over to this, and you will see U, go over to A for the second one, and then we see C. So this should be tyrosine, which we can see over here. And this is generally how the genetic code is read and translated into amino acids. And that will give rise to amino acids in a sequence that will make the primary structure. And you will see here we have an amino acid with the actual chemical molecules drawn out here, and these will give rise, based on what amino acids are in what order, will give rise to either alpha helices or beta sheets. And then from there, these secondary structures can make tertiary structures, which are even larger, and then larger subunits can come together to form quaternary structures. So this slide does a good example showing how regulation of transcription occurs. This just shows a generic cell, and within the nucleus, we have DNA that can be seen, and it is showing in this image where it's wrapped in histones, which is very important for control and regulation, and where it's open, it allows different proteins or RNA polymerase to bind, and it can actually make those mRNA templates, which can be translated and turned into amino acids that would build proteins, which could include more histones as shown in this image. So inside of a cell's nucleus, as shown by this darker image here in the center, is where all the DNA is contained. DNA is contained within 23 chromosomes. 1 through 22 is known as autosomal, and the 23rd chromosome is an X from the mother and either a Y or an X from the father. And these 23 chromosome pairs all contain the genetic information that is within an organism and is needed to be expressed for them to maintain homeostasis and to be able to produce and have offspring that can carry forward their genetic information. So we've covered the DNA double helix, how it contains the code in codons in groups of threes. These can be translated, transcribed into RNA, but they are also modulated by proteins such as histone, shown here, and they are wrapped and coiled very tightly into large chromosomes. So now I'm going to cover the pedigrees and patterns of inheritance. Pedigree, you can see shown over here on the right. Typically, the mother or mom is shown in a circle, and fathers or dads are shown in squares. And what this is showing, and the reason I wrote 23 chromosomes, is because each parent, a human being, has 23 chromosomes. But in gametes, which are the egg and sperm, they are reduced because humans are typically diploids that have two sets of chromosomes. Gametes only have one set, which is represented by this one N. So one N from the mother and one N from the father will combine to create 2N. So there will still be 23 chromosomes, but there will be duplicates. There will be two pairs, and those pairs are coming from each parent. And that will form the zygote, which will be a unique combination from the parent's DNA, and also genetic mutations could occur as well. So parental DNA is inherited into the offspring. So the mother, shown here on the left, will provide 23 chromosomes, and the mother will only be able to provide the X chromosome because her being a female, she does not have a Y. And also, the mother's egg will also contain the mitochondrial DNA, which is an organelle that exists inside human cells and provides energy. So the father, he will provide also 22 chromosomes here, and his 23rd chromosome, if it's an X, will produce a female offspring, or if it's a male, it'll produce if it's a Y chromosome, it'll produce a male offspring. So here is a zygote, and this is basically just a child. And I like how this is shown here from 23andMe. We have this one pair, so you can imagine that the mother's providing this one here, and then the one that's to the right is being provided from the father, and it just goes down the line. So there's a pair of each one that can be used for genetic templates to make proteins in the body. And then also you can see down here, since this is an X and this is a Y, this is the DNA results from me, and me being a male, I have to have this Y chromosome, but the mitochondrial DNA that I have here came from my mother's side, which also came from the egg. So these are ways that DNA can be inherited into the offspring, and we'll focus more on pedigrees now that I think this covers in detail how the DNA is transmitted. So pedigrees and patterns of inheritance, occasionally things happen which are abnormal mutations. So typically a parent will pass a genetic defect or a genetic mutation to an offspring, but sometimes both parents are completely wild type, healthy normie individuals, but whenever they have a child, a mutation will occur at random. And I'm going to go in more detail about this later. So patterns of inheritance, this is a very busy slide, but this is a good website to start with to understand the patterns. And this is more information than we need here. So what I'm going to focus on is this. The pattern of inheritance in autosomal dominant diseases such as Huntington's disease, and also autosomal recessive diseases, which happens in sickle cell disease and also cystic fibrosis. And this image here in the bottom is a larger pedigree, which is shown, and typically the half shades show that an allele is present, but the individual is a carrier. And typically whenever it's drawn in fully, this indicates that the individual is affected with a disease. So the dominant form requires there to be one mutation, and the recessive requires there to be two mutations. So here we can see that the dominant has a mutated gene, and this would be as in the case of Huntington's disease. So an affected parent would also have a 50% chance of affecting their child if they inherit that gene. And the same for unaffected as well. And if you follow these lines, you can see clearly how this goes together. So the autosomal recessive inheritance is a little bit different. So this one is the carrier isn't necessarily diseased, but they could have a diseased child if two carriers have a child together. So the karyotype of 23 chromosome pairs and genetic disease example overview. The Huntington's disease, from what we've seen here, I just want to point out it would be on chromosome 4, and it's also identified that it's in the P arm or small region, and the P indicates it's the small arm, and the Q indicates it's the longer arm of the chromosome. So we would say in this location here. If there is one genetic mutation in the HTT gene where it repeats, then this will cause a mutation that can be passed to the offspring, and having one present would make it a 50% chance the child would inherit the disease. Now, with cystic fibrosis, it's autosomal recessive, and it would occur on one of these arms here. And if you had one, it would just be a carrier. But if you actually had two spots affected, then you would be afflicted with the disease cystic fibrosis. A similar inheritance pattern also occurs with sickle cell disease here on chromosome 11. Whenever you have one spot affected, you would be a carrier. But if you have a spot affected here and a spot affected on the other side and the same allele, then this will cause the disease to occur. So dominant requires one mutation, whereas recessive only requires two. This pedigree here is very small and shows basically what happens. Every generation is affected. So one person has Huntington's disease, the other does not. There's a 50% chance that the offspring will also have the disease. And since it's autosomal, it doesn't matter what the sex of the child is. It can affect them equally. And on the right here, we can see autosomal recessive inheritance, where we have two people who have the recessive, they have the mutated gene, but it is recessive, so they don't exhibit any sign of the disease. So here we would have a 25% chance that everything would be okay and normal. There's a 50% chance that the child would be a carrier, and there's also a 25% chance that the child would have the disease. And if you do have the disease, when you have kids with someone else that does not have any genetic mutations, there is a 50% chance that it is going to pass to the child because you only have two chromosomes that are mutated to pass on. So there has to be carriers. So Huntington's disease was one of the first diseases to be identified that a gene was involved. This is more noticeable because if the parent has the disease and the child has the disease in the next generation, obviously something has to be occurring. It was also one of the first located, and it was identified to be the HTT gene, and this gene is located on chromosome 4 on the short arm. And in healthy individuals, there's a number of repeats of these nucleotides, the CAG, as we can see here. And whenever this repeats, it will encode for the amino acid glutamine. And in healthy individuals, there's, say, maybe 27 of these repeats. But whenever this number begins to exceed to 36 and beyond that, then the disease starts to be noticed and you start seeing symptoms related to it. And what this does is it will cause defects in the brain, so neurons will start to become damaged, and motor coordination and memory and cognitive function will all begin to decline just because of this trinucleotide repeat that continues happening to an excess of 36 or more. So now I want to go into the pedigree of autosomal recessive patterns. and this is the order at which they follow. Two people with the recessive gene for the same disease can have children, 25% chance the child will be healthy. There's a 50% chance the child will be a carrier, and there is a 25% chance the child will have a disease. So cystic fibrosis, or CF, as it can be abbreviated to, is also an autosomal recessive inheritance disease, which follows this pattern. What happens in this disease is that the gene, the CFTR gene over here, which we see the sequence for, is important for encoding a chloride channel protein, and that is located down here, which we can see this channel. And what happens is, well, one of the most common cases, and about 60%, is we see this right here, deletion at position 508, where a phenylalanine is removed. And whenever this happens, it causes an error in the pocket of the ATP binding domain. So ATP will typically bind here. It will identify the amino acids within this region, and once it locks into place, it can break its phosphodiester bond, releasing energy and allowing the chloride ion channel to function properly. And this is important for many things, and whenever you inherit one, you may have some malfunctions with these channels, which could increase mucus or cause other issues. But when you inherit two mutated alleles in the same region, then what will happen is these ion channels will become very ineffective, and it will result in a lot of mucus building up inside the body. And it could even cause a point where your lungs would have to be removed and replaced with a donor for you to be able to survive, because otherwise you're going to have difficulty breathing, and without any medication or any intervention, the lifespan is reduced to about 30 years. So pretty terrible disease, but being able to identify the mutations can at least let people know in advance if they are going to be at risk passing on this recessive disease to their child. Sickle cell disease is also an autosomal recessive disease. So typically wild type people have hemoglobin A, and hemoglobin A is the oxygen carrying protein inside red blood cells, and usually these are just free floating, and they're able to take oxygen from the atmosphere and transport it to the cells of the body. But in the 6 codon region, where one of the amino acids is GAG, that makes glutamic acid, which is a polar amino acid that wants to face outward, it becomes mutated and changes into GTG, and this transversion mutation from an A to a T changes to a valine amino acid. So now we have this amino acid substitution from a polar glutamic acid to a non-polar valine. And whenever you get this inherited from both parents and you have two valines, they will begin to aggregate and form these long chains here, and these become insoluble and they will form really long rods, as we can kind of see here. And those rods will form into what we see as a sickle-shaped cell. This is a good pedigree too that shows how this passes. The reason they're using this beta here is because the sickle cell traits that cause sickle cell disease, sickle cell anemia, and beta thalassemia are all occurring on the beta chain of hemoglobins. So here we have HB, where we have the beta A as normal wild type, and then this person has a wild type one here, BA, but then they also have the beta sickle allele, which was shown on the previous slide. So if these two families have children and their children both contain the mutation, and then it's passed on to the next one, from here we see a carrier and a carrier who may not know they have sickle cell disease unless they're in extreme conditions where they're low on oxygen, dehydrated, and maybe at high altitudes. Would they start to see sickled cells and have problems? But when they have children, their children are going to have a 25% chance of being free of the sickle cell allele and have no risk of passing it to their children, or there's a 50% chance they will still be carriers and they can still pass on this allele to their children. but unfortunately there is a 25% chance that the child born will have sickle cell disease, and in this case it would be sickle cell anemia because it's the beta SS in the 6 codon region we just saw for valine, and this person would be homozygous with the mutated alleles and have the disease.